Hello everyone. Today we will be talking about human-animal chimeras, specifically human-pig xenotransplantation. To put it simply, xenotransplantation is the process of grafting or transplanting organs or tissues between members of different species. In our case, scientists are hoping to grow human organs in pigs that are then used for transplants. However, currently these human-pig chimeras are not allowed to develop past the fetal stage. Using pigs for organ transplants sounds odd, and you may be wondering why go to this extent. Well, currently about 120,000 people in the United States are waiting to receive an organ transplant, but due to shortages, only about 30,000 transplants are performed. As a result, approximately 22 people die each day waiting for a transplant. You can already understand the potential of human pig organ transplants, but we have to consider the restrictions, the dangers, along with the positives. Now, let us talk about the history of xenotransplantation.
Hello there, this is Pen. Here I will briefly introduce the history of xenotransplantation and why the research of human-animal chimera, especially human-pig chimera, plays an important role in organ transplantations. The very first serious attempt of cross-species organ transplantation was recorded by scientific literature in 1905, when a slice of rabbit kidney was given to a patient with chronic kidney disease. That experimental treatment failed without surprise due to immunological rejection. Later studies about the immunological basis had greatly reduced scientific interest in xenotransplantation. Many attempts were made afterward, but all ended up with failure. However, it is noticeable that in 1984, an American girl known as Baby Faye died 21 days after receiving a Dublin heart. Although the treatment did not succeed, the story grew so big and rang the bell for the organ shortage at the time, which promoted the progression of xenotransplantation. As for preclinical research, xenotransplantation of human cancer cells into immunocompromised target animal is a research technique often used for oncology research. Clinically, organ transplantation is often considered as a potential treatment for end-stage organ failure. Although xenotransplantation has massive potential in curing disease, it also raises concerns regarding potential infection with unrecognized agents and possible subsequent transmission to the human population. Human-animal chimeras are often considered as a potential organ source for xenotransplantation to humans. Although chimpanzees were originally considered the best option, pigs are currently thought to be the best candidates for xenotransplantation. Besides being easy to breed, pigs have organs that are anatomically comparable in size. There were also successful treatment sources from pigs in record. Although the development of human-animal chimeras and the application of xenotransplantation seems to be very conservative in terms of disease treatment, they together have faced technical concerns since the very beginning of their history, which will be discussed next.
Genetic engineering of pigs for organ transplant into humans is necessary for the transplant procedure to be successful. Attempting to transplant an unmodified pig organ leads to rejection within hours, due primarily to the carbohydrates on the donor cell not matching those of the host. That being said, developing and establishing methods for the modification of pig DNA was a must. The most successful and currently researched method of inducing gene edits revolves around injecting bovine zygotes with CRISPR-Cas9 systems. Zygotes can be edited using CRISPR with higher efficiency than normal somatic cells. The zygotes are then grown into fetuses for 32 to 42 days, at which point the nucleus from the fetus are harvested and used to create the next generation of modified pigs, using a process known as somatic nuclear transfer. Somatic nuclear transfer, or SCNT, involves taking a nucleus from a donor's somatic cell and putting it into a donor egg cell that has had its nucleus removed. The new hybrid cell is then shocked and will begin dividing into a new organism. Usage of this process allows researchers to save time by rapidly decreasing generation time between modifications. Many of these genetic engineering efforts in pigs have focused on protecting against transplant rejection. This has taken the form of knockouts for genes that code for pig-specific identifying carbohydrates, as well as the introduction of human biomolecule genes. While genetic engineering efforts have worked to put off the eventual organ rejection, other methods are being investigated to work alongside transgenics to improve host tolerance to donor organs. The two methods are mixed chimerism and thymic transplantation. Mixed chimerism involves grafting in bone marrow from the donor along with the donor organ. This helps the host tolerate the donor organ, but carries the risk of the host attacking the introduced marrow. Another method involves transplanting the donor's thymus along with the desired organ. This prevents killer T cells from attacking the donor organ by growing donor T cells inside the host, and induces tolerance. Despite the many hurdles to overcome, xenotransplantation research is making rapid progress. According to Dr. Klassian, the Chief Medical Officer at the United Network for Organ Sharing, clinical trials of this burgeoning technology are only 1 to 2 years away. With the future of xenotransplantation already on the horizon, it is apparent that we as a society must consider the moral implications of creating these human-animal hybrids.
There are two major arguments in favor of xenotransplantation. The first is that xenotransplantation provides a solution for the lack of organs available to people currently on the waiting list for organ transplants. According to the Health Resources and Service Administration by the U.S. government, 120,000 plus people in the United States are currently on the waiting list for organ transplants as of September of this year, with 22 of these people dying every day. As we can see on the graph to the right, there are many more organs needed than received each year. This brings up a serious ethical issue, since these people would be able to live much longer if only there were enough organs available for transplant. Xenotransplantation can solve this issue by providing more organs for transplant, since the pigs meant for producing human organs can be bred on large scales. The second argument for xenotransplantation is that it also reduces the risk of donor-to-patient disease transmission. According to a journal written by White and her colleagues, disease transmission is rare for transplants. However, when transmission does occur, it often leads to mortality. This is because these diseases are often quite serious, such as HIV and hepatitis B and C. Patients should not need to worry about the risk of getting a disease when receiving an organ transplant, and xenotransplantation can reduce this risk significantly, since the pigs that are farmed for transplants will be kept in sanitary conditions and screened for potential diseases regularly.
Despite all the benefits that xenotransplantation provides, it is not without risk. Transplants from pigs face obstacles from the innate immune system because of their differing antigens from humans. This is especially important in the rejection response because innate immune cells can also be activated by xenografts in the absence of antibodies. This means that even though the body isn't actively rejecting the organ, the immune system is passively trying to remove what is considered foreign in the body. Another issue is the uncertainty in how the immune system will react, even if we consider the use of genetic engineering. This was shown with the gal carbohydrate in pigs that caused acute rejection, but when removed, made a delayed xenograft rejection which occurred days or weeks later. The fact is that not all variables are accounted for in genetic engineering, since there are many obstacles to xenotransplantation that are not immediately noticed. The dangers of a delayed rejection can be fatal, and is why clinical trials should not be available until the risk or at an acceptable level. Even if we resolve the antigen issues, the possibilities of diseases transferring from animals to humans, as mentioned previously in another slide, which we refer to as zoonosis, can occur because of the patient's immunocompromised immune systems. This is further worsened by the possible risk of the pathogen spreading to the community, which is much more unlikely but should be considered. The reason why zoonosis is so dangerous is because the immune system of a xenotransplant patient would be purposely weakened to prevent organ rejection, but this leads to them being more vulnerable to pathogens instead. However, as mentioned before, the likelihood of zoonosis is pretty low, and the possibility of it spreading and becoming an epidemic is even lower if precautions are taken beforehand. This leads to another issue that is on ethical grounds. Whether we should monitor patients for the rest of their lives, which intrudes on their privacy to ensure that the transplant organ is relatively safe for both the individual and those around them. But is it justified to ensure safety at the expense of personal privacy, despite the ethical concerns? Perhaps giving patients a chance at life is better than leaving them with no opportunities at all. And that's why, even though we have so many different disadvantages, those should not limit what we should do with xenotransplantation in the near future.
Hi, I'm going to talk about our choice and why. Based on the arguments talked about before, we think the benefits of xenotransplantation outweigh its disadvantages. One big reason is it will solve the organ shortage. As we are already known, over 120,000 people in the United States are waiting for organ transplantation, and only a small portion of them can obtain suitable organs. The figure on the screen shows the large disparity between the number of patients on the waitlist for transplantation and the number of available organs. Currently, there are three possible sources for organs. The first one is organ from deceased donors, and it is what we are using right now. However, we cannot expect more people to die in the future, so there is limited potential for more organs, and we should consider new methods. Second, bioengineering of transplantable organs. This approach uses stem cell as the source to produce animal or human organs, but there is a problem. It needs stem cells from specific potential recipients to prevent antigenic differences. So this approach is less practical for the large need of organs. The third one is xenotransplantation. One advantage of this method is there is a limitless supply of organs from animals. And animals like pigs are similar to humans in size, psychological, and anatomy. So xenotransplantation has the biggest potential to cover the large demand and easier for scientists to approach and make it possible in the future. Another reason we support xenotransplantation is there is a big potential to solve the problem. Although the problems such as unexpected immune response and transmitting endogenous retrovirus are existing, scientists are working on solving them and already have some achievements. For example, New et al. Generated PERV inactivated pigs by CRISPR-Cas9, and PERV inactivation is effective in preventing cross-species viral transmission. All in all, with the continuous development of transgenic pigs and improvement of immunosuppressive methods, the problem can be solved in the future. For the action and consequences, xenotransplantation has excellent prospects, so the research in this field should continue. If the study could be continuously going on, more organs will be applicable for xenotransplantation, as the figure shows here. In the near future, the cell and small tissues like brain cells and eye tissues can be transplanted to humans, and the transplantation of solid organs like the heart and the kidney still needs several years to solve the problem. Xenotransplantation still has a long way to go, and there are still many technical problems and ethical concerns that must be solved. But this is an area worth challenging because it will save many people's lives. In the future, xenotransplantation will truly enter the stage of clinical application and will eventually alleviate the organ shortage.
In conclusion, human-animal chimeras, specifically human-pig xenotransplantation, have a great potential for providing people that need these transplants with a higher chance for survival and allows for alternate treatments. With the implementation of this research and technology, not only will this help remove the need for donor organ waitlists, but it could help us further the understanding of the human immune system better and what we can do to eliminate and treat diseases. Although this technology has a great potential to be beneficial, it is still a relatively new field with possible consequences that we do not yet know about or understand. Some such concerns that people have are the possibility that those with these transplants could be more vulnerable to zoonotic diseases and could spread them. If this technology is to be implemented, I would imagine it would go under extensive testing and research before widely used. Even so, we are for this technology and believe that its potential benefits outweigh any of its pitfalls. We do not expect this technology to be implemented anytime soon, as it seems to be in its relatively early stages.